Hello, this is the first of several modules on DNA technologies, including cloning and other aspects of recombinant DNA and genetic engineering.
Our technological prowess in manipulating DNA and doing cloning are dependent on our ability to recombine DNA pretty much at will, and uses many of the enzymes of replication, as well as a few others we really haven't had a chance to talk about. So let me introduce those right away. One of them is reverse transcriptase. There are retroviruses whose genomes are actually RNA, not DNA. These viruses infect eukaryotic cells in this manner. They fuse their membranes with the cell. Their RNA genome enters the cell and then eventually enters the nucleus, where an enzyme, reverse transcriptase, reverse transcribes a complementary DNA copy called cDNA, complementary DNA or copy DNA. This eventually becomes double-stranded and replicates in the infected cell nucleus to direct the reproduction of more viruses, and that's, of course, the basis of infection. Reverse transcriptase then became one of our other cloning tools. Another enzyme, or group of enzymes that's very important in our ability to manipulate DNA and recombine fragments of DNA pretty much any way we want to, are restriction endonucleases. These are enzymes that were evolved in bacteria as a way to protect the cells from infection by bacteriophage. The DNA of the virus, once it got in the cell, if it was detected by a restriction endonuclease, would be cut up before it had a chance to do its damage. Restriction endonuclease is named because it would restrict phage growth, and then endonuclease means a nuclease that attacks a DNA polymer somewhere in the middle of the chain. As you will see, restriction endonucleases recognize, most of them that we use, recognize very specific sequences of DNA, usually very short, 6 to 8 bases, or 4 to 8 bases actually, 4 bases long to 8 base long sequences, that they then bind to and cut. So here's our restriction endonuclease digestion of a piece of DNA at a recognition site, at one of these, in this case, a 6-base recognition site. And as you can see, the digest was unequal. That is, it occurred at 2 different positions on either of the 2 strands of the double helix, generating what are called sticky ends. For the bacterium, that was enough to inactivate the bacteriophage DNA. We can use this phenomenon, the existence of restriction endonucleases, to enable our ability to recombine DNA at will. That is, any fragments of DNA that we wish to recombine, mix together and make a single strand out of, we can do it. With that introduction to 2 specific enzymes that are among our tools, along with DNA polymerase and DNA ligase, as you'll see, we can talk a little bit more about DNA technologies and what they're good for. We're going to look at cDNA cloning. That's cloning of these copy DNAs that we can make out of messenger RNAs using reverse transcriptase. We're going to look at genomic cloning. Now that's cloning of chunks of chromosomal DNA, usually a good deal larger than a cDNA, which after all is just essentially the size of an mRNA, right? We're going to look at something called PCR, or polymerase chain reaction. These are major tools in our toolkit for doing gene structure studies, regulation studies, and gene evolution studies. We'll talk about how DNA is sequenced, and I'll mention at that time genome projects and how it connects. We'll talk about something called microarrays. This is already now a technology of some 10 years standing, which allows a genomic approach to studying genes, gene structure, gene activity, because it allows us to screen, it says multiple genes at once here. In fact, almost the whole genome can be screened at once and in a very small space, and I'll try to drive that home when I show it to you. I don't know that we'll get a chance to look at how sequence databases are being used, but if you've heard the term bioinformatics or bioinformatics revolution, then you have some notion of what I'm talking about. If you start accumulating large amounts of DNA sequence data, it's going to be necessary to store the data, retrieve the data, and make comparisons with the data. And we have wonderful DNA sequence databases to house our sequences, compare any new sequence with anything that's out there in the database, and decide if you have something brand new or if you have a DNA sequence that resembles an already existing sequence, and so on. We may have a chance to talk about the promise of gene therapy beyond this point, but clearly if we can engineer a healthy version of a gene to take the place of a mutant version of a gene, say for insulin or for growth hormone or for any molecule that is a problem in an individual that carries a mutation, theoretically it might be possible to cure a genetic disease. And that involves genetically engineering cells to produce what you want. In a nutshell, that's what we are aiming for in an application of DNA technology. So I said that you can recombine DNA from any two sources, any two organisms that you wish. You simply cut them with the same restriction endonuclease, and then you mix the DNA from the two sources in this fashion, and you add DNA ligase, which will catalyze the stitching together of DNA fragments, and you end up with recombinant DNA. So in this example, we took a plasmid, which is a piece of circular DNA independent of a bacterial chromosome, but often housed in bacteria. These plasmids reproduce independently. You can look up plasmids in the textbook on your own, but basically they're little circles of DNA. If you cut them with a restriction enzyme, you get that one that produces sticky ends, and then cut some other DNA with sticky ends or produce DNA with the same sticky ends. You can combine them. The complementary sticky ends will we say, anneal. They'll come together. They'll form H bonds. And then if you use DNA ligase, you can make whole circles that now contain the various fragments you've added to the linearized plasmid DNA. So here's one blown up a little bit, just showing the structure of this recombinant. So we have mostly plasmid, but it's been recombined with some foreign, in this case, double-stranded copy DNA, right, that was made with a reverse transcriptase, to which has been added these sticky ends, which we can do chemically. So if we use this technology, we can make a cDNA library. What is a cDNA library? It is a compilation of as many messenger RNA molecules as a cell can produce, cloned into plasmids, and then distributed into E. coli cells or some other bacterium, but usually E. coli, such that each E. coli cell contains only a single version of the plasmid, which it then reproduces to clone a specific sequence. So let's make a cDNA library out of messenger RNA isolated from reticulocytes. So the first thing you do is you isolate total RNA from reticulocytes. Reticulocytes are the predecessors in our bodies of erythrocytes. Remember I told you in an earlier module that chickens have red blood cells that have nuclei, but our red blood cells lack a nucleus. So the precursor to our red blood cell is a reticulocyte, and the reticulocyte is the place where the RNA for globins are transcribed and the globin proteins are produced. And our erythrocytes live about 60 days, carry out the functions of oxygen binding and delivery, and then they die. And they have to be replaced by new reticulocytes, which then lose their nucleus and then function as erythrocytes again. Well, we know how to get reticulocytes out of mammals, ourselves included. And if we isolate total RNA from a reticulocyte, which is, of course, still producing transcripts for their various proteins, we can put that RNA onto a little column containing gray stuff. Now, the gray stuff is synthetic. It's basically a bead to which has been attached a string of T's. You should remember that one of the things that happens to 99.9% of eukaryotic messenger RNAs is that as part of the processing, they get a poly-A tail of up to 200 A's. So if you have a solution of beads with oligo-DT or poly-T stuck on them, when you add RNA, the poly-mRNA is going to bind or stick to those T's, and all the other RNAs are going to flow through the column. So here we have an illustration off to the left of polyadenylated mRNA sticking to the oligo-T's on the beads. So if that happens to the RNA you add to this column, what you collect then you can discard. That's non-poly-A RNA, which means it's not message, right? Mostly not message. It'll be the ribosomal RNA, the transfer RNAs, and so on, but it will not be message. After throwing away the stuff you don't want, you add a special buffer, and off comes, basically you're destabilizing the H bonds between the T's and the A's, and off comes the polyadenylated mRNA. We say it's eluted from the column. And so now we have poly-A mRNA in this tube over here, in this test tube below the column. If we add reverse transcriptase to that solution along with 4 deoxynucleotide triphosphate precursors to DNA, we will synthesize red blood cell or reticulocyte cDNAs, copy DNAs. The process of making what starts out as a single strand of cDNA into double-stranded cDNA is a bit complex, but it involves reverse transcriptase and some primers that go in the opposite direction. Again, it's a DNA synthesis reaction. Suffice it to say that you end up with double-stranded cDNA. I showed you this process earlier, a few slides ago. I'll show it to you again. So here we have a plasmid on the left. We're going to linearize it. Remember, plasmids are circular double-stranded DNA molecules that exist in bacteria apart from the actual chromosome. They're a lot smaller. If I treat with a restriction enzyme that cuts this plasmid open at one site, I end up with a linear piece of DNA with sticky ends. Now, I've got these cDNAs, and I don't really want to cut those, but I do want them to have sticky ends. So what I do is I synthetically produce what's called a linker, which has the same sequence of bases that would be recognized by the restriction enzyme I intend to use, and I ligate them to the cDNAs, and I get this array of cDNAs, each with synthetic DNA on either side that contains an endonuclease site, a restriction endonuclease site. So I've used DNA ligase to stick them together, and hopefully that's what I get. And now if I treat this material with the same restriction enzyme, I will indeed get double-stranded cDNAs with sticky ends. Now I can add these two end products together, like so. And the set of sticky ends on the cDNAs, which I created synthetically, and those on the plasmid, which I created simply by digesting the plasmid, will come together. And now I can add DNA ligase again, and I get plasmids with inserts. And if I do this in just the right way, I get plasmids with all the different possible cDNAs representing all the different original RNAs that were, or messenger RNAs that were isolated from these red blood cells. These are then all recombinant plasmids, each with, we say, a different insert cDNA, a different insert. So now we want to make a cDNA library. So the library is ultimately going to be E. coli cells, each of which contains one but no more than one of these different insert-containing cDNAs. So we mix the two together under conditions which allow transformation. You remember transformation as the very early experiment of Griffith using Streptococcus. He discovered that Streptococcus, under the right conditions, will take up DNA and incorporate the DNA into the cell. Well, you can do the same thing here, only this time the plasmid is being taken up. It's done under conditions where one plasmid ends up inside of a cell. The cells are not healthy if they take up more than one plasmid, and so those which take up two plasmids will generally not survive. So what you end up with is a tube full of E. coli cells, each with a different plasmid in it. Now there's obviously redundancy here, right? There are many cells that have taken up versions of the same plasmid or recombinant plasmid. But the bottom line is there are millions of cells here, and among those millions of cells will be at least a few that have one cDNA, recombinant cDNA, and another a couple of 100,000 cells that will have another cDNA. That's your library. Now the issue, of course, is how do you pick out of that library a clone that has something that you want, that is a cell that has something that you want, that you can clone a lot of. So how do you find, in this case, a Globin cDNA clone? Well, bacteria are grown on agar dishes, shown here, and so what you do is you spread the cells of your library onto an agar plate, and colonies will grow from single cells. So what you've done is spread the cells on an agar plate under conditions where there are not too many cells on every plate, say a few hundred. And so you get a few hundred colonies. So now the library has been displayed on an agar plate. Again, the library represents a subset in this case. If you have a few hundred colonies and you expect a few thousand messages, the library represents a subset of the transcripts that originally came from the red blood cell at the time of extraction in the form of recombinant cDNAs. So now you have grown this plate, that's the original plate. What it's done is to place a filter on top of the colonies to produce a replica. So you're going to put the filter on top. The filter is going to pick up a few cells from every colony, and then you take that filter and you turn it right side up, colony side up, and put it on a fresh plate. And over time, growth in that dish will produce a mirror image of the colonies that were on the original plate. That's the replica plate. What do you do with that filter? Well, it's got a bunch of bacteria on it in the form of colonies that are now a replica of what you had before on the agar plate. But what you're going to do here is you're going to burst open the cells, lyse them, and raise the temperature or change the chemistry of it so that the DNA that is in those cells, first of all, denatures, that it becomes single-stranded, and then sticks to the filter in situ, meaning in place. So wherever there was a colony, there will be the DNA from that colony stuck on the filter. So that's lysis along with denaturation. And then you're going to expose that now single-stranded DNA arising from each colony to a radioactive probe DNA, which in this case is going to be a radioactive Globin probe, and I'll show you that in a moment. So we've done the lysis, we've done the denaturation. You no longer see colonies, but what you see is in reality, you see a sort of a pale yellow remnant of a colony, of each of the colonies, which contain the DNA that came out of each of those cells, but didn't go anywhere. It stuck to the filter right in place. So there's DNA now where there used to be cells. We soak this filter in denatured probe, that is radioactive probe. What's the probe? Well, it's really going to be either a denatured piece of DNA or DNA that's single-stranded to begin with, that is going to be a complement of, in this case, a Globin gene, or part of a Globin gene. So the idea is that the probe is going to seek out and find complementary sequences in the recombinant plasmids. The only place that those recombinant sequences could have come from is cDNAs that were essentially copies of mRNA for Globin. Well, I'll show you the results in a second, but how do you make this probe in the first place? That's a cart before horse question. In the early days of cloning, we really had very little to go on. We were able to do amino acid sequencing, for example, of various polypeptides and proteins. So here's an amino acid sequence of a Globin polypeptide. So we can look at that and we can say, what DNA or RNA would actually code for this? Well, here is, in the form of DNA, the possible code words that you could use to encode a portion of this polypeptide. And that's basically determined by going back to the genetic code dictionary, also in this case to an amino acid dictionary, so you know what Q, F, A, G, K, and D are. K is lysine, I remember D is aspartic acid, G is glycine, A is alanine, F is phenylalanine, and I forget what Q is, probably glutamic acid or something like that. And you go to the genetic code dictionary, and you see, Q is encoded by CAA or CAG. Phenylalanine, F is encoded by TTT or TTC. Remember that some of the amino acids have only 2 code words. Some have 1 code word, some have as many as 4, and those are shown here. So you take all those coding possibilities, and you synthesize. What we did is we went and paid someone to do it for us. We had an oligonucleotide produced, which was really a mixture that had all of the possible sequences that could code for this amino acid sequence QFAGKD. Turned out that was 128 different oligomers. In other words, the one that you see here is CAA, TTT, etc. We had another one that was CAG, TTT, etc. along the top line. And then we would have CAA, TTTC, GCT, etc., and so on. So if you figure out all the ones you can get, it's really, let's see if I did it right. It's 2 possibilities at the left end, CAA or CAG, another 2 possibilities in TTT and TTC, and so if you just multiply these numbers together, it's 2 possibilities times 2 possibilities, that's 4, times 4, that's 16, times 4 is 64, times 2 is 128. That's where I got the 128. If you have questions about this, you can do this in your head, or you can contact me and I'll explain in more detail how you get 128 different sequences that could have encoded this region of the polypeptide. So we made this mixture and we made it radioactive. There's a way you make it radioactive. So that's our probe. It's a mixed probe. And when we soaked our filters in solution containing the probe, several of these colonies in several of these replica filters turned out to contain Globin genes. Let's look at what happens. This is how you make an autoradiograph. We have already soaked this filter in radioactive probes, so if there is a Globin gene or Globin cDNA in the DNA from any of these filters, it's going to be on that filter. I'm going to put that piece of film right on top, We're going to wait a while, expose in other words, and then after that we go into a dark room basically, and we take the film out and we develop it. It's like developing an X-ray. And lo and behold, there's a black spot on the film, and if we have properly prepared this, we can tell that that black spot came from a particular colony, and you can see that in the illustration here, came from one of those original colonies. Now you'll know why we have done this on a replica filter. That positive signal can be traced back to the original agar plate, where there is a real colony still alive, which contains a plasmid, which must have a recombinant plasmid containing a cDNA, which was derived from a messenger RNA that had been part of what was isolated from the red blood cells, from the reticulocyte. So we pick the clone, the Globin cDNA clone, which is to say we pick some cells or pick that colony from the original dish. We can grow it in a liquid culture. We can extract the plasmid DNA. We can do a lot of things to it. The two things that are often done when this procedure is followed is we sequence it. We find out the sequence of the entire recombinant insert DNA, the entire bit of Globin DNA that we have. And then we can use that rather large chunk of Globin DNA to screen a genomic library, and I'll show you a genomic library in another module. And I'll also show you DNA sequencing in another module as well. The key here is that if you think about what we just did, the probe was, I don't remember, you can count them up, I think 17 nucleotides long, and it's being asked to find a 17 nucleotide complement in recombinant cDNAs, and it does so very effectively. Small probes like that will not find clones in a genomic library because it's looking in a much bigger haystack, if you will, for that needle. But if you have a bigger probe, which is several hundred bases long perhaps, that is a complement to a gene in the genome, in a genomic library, you can pick a clone of an entire gene. Remember that a cDNA is just a portion of the gene that's been transcribed, whereas the entire gene will include lots of other information. If you pick a clone out of the genomic library, it will include things like the region of the promoter, which is not transcribed. It will include perhaps regions of DNA that regulate transcription of the gene. Hence the need to isolate cloned genes, not just the cDNA copies. But the first step is often making cDNAs from isolated mRNA in order to create a cDNA library, screen it, pull a clone, and use it either to sequence it directly, just to know the sequence, or use it as a probe of a genomic library. So that brings us to the end of this presentation.